Okay, I think we can get started now that we have a quorum. So thanks everyone for joining the OpenTrons webinar series. Today we have two great speakers, OpenTrons CEO John Brennan-Badal and our scientific director, Kinnary Watson. And they'll be talking about some advances we've made with our OpenTrons Flex that really changed the game in terms of making NGS library prep more powerful, more accessible. So just before we get started, one or two administrative type notes. This webinar is being recorded and it will be made available on the OpenTrons website afterwards if you want to review the recording. And we'll also send it to you via email. Everyone is on mute as is normal, but you can ask a question at any point during the presentation using the Q&A box at the bottom of your screen. Just type your question in there and we'll put the question to John and Kinnary at the end of the talk. And so with that, I will hand it over to John to introduce the topic and take it away.
Okay, great. Thank you everyone for joining. And so we're here to talk about reducing time to science, how accessible touchscreen enabled automation makes NGS library prep plug and play. So it's obviously a lot to unpack. And so we'll talk a bit about OpenTrons' approach to automation, then talk a bit more specifically about how OpenTrons automates NGS library prep, and then discuss data from a wide range of sequencing technologies. And so I'm John Brennan-Badal, CEO of OpenTrons. And Dr. Kinnary Watson, do you want to introduce yourself?
Yeah, I'm Kinnary Watson. I'm the scientific director at OpenTrons, who leads the internal application development. So glad to be speaking with you all today.
Great, thanks. And so we'll jump into OpenTrons' approach to automation. So first, just a bit about our mission and how that I think differentiates us from many other automation companies. We as a company, we're really focused about empowering all scientists. It's our view that so many companies in this industry really focus on making very kind of expensive, inaccessible solutions that really only serve a very small subset of labs out there. And so we're really focused on making easy to use tools that are highly affordable so that many more labs can have access to laboratory robotics. And in doing so, accelerate the amount of innovation and discovery that's possible. Let's jump on to the next, slide. And so to that end, we focus on being radically accessible, which is to say that highly easy to use solutions that are affordable and can be used, quite frankly, right out of the box. Second, we're really built for innovation. And so we have an open tech stack that allows our customers as well as partners to integrate and build on our platform. And then lastly, we're ready to scale with you. We have our systems are highly modular, can be easily reconfigured, and very easy to scale. I won't, read through all these kind of quotes, but just to kind of call out, because of that, our unique approach, many customers, whether it's academic labs, biotech startups, or even large kind of established life science companies, really have found a lot of success and really love, in particular, our new Flex robot. Just to give you kind of a sense of, OpenTrons, as a whole, we've deployed and installed over 10,000 robots worldwide, deploying, multiple robots every day. We employ over 250 people and serve customers in over 40 countries around the world. I'd be remiss if I didn't talk a little bit about our Flex robot, which we brought to market last year. This really is, I think, a game changer for OpenTrons, and it's really built to enable automation of complex workflows like NGS library prep. While still maintaining a high degree of affordability, it has a lot of the capabilities that you might expect out of far more expensive systems on the market. And so it's, like the OT2, a modular benchtop liquid handler. You can configure it with single, 8, and even 96 channel pipettes. It has a gripper that you can be using to transport plates and labware to any kind of position on the deck. Has a range of modules, including the thermocycler, heater shaker, mag block, etc. And enables kind of bundled workstations for applications like NGS. And with the on-device display, as well as other kind of significant kind of improvements, it's designed really to maximize walkaway time and minimize runtime for complex activities like NGS library prep. To say a little bit, more, this on-device display really simplifies the user experience for working and interacting with the robot. With that on-device display, you can run automated calibration, processes on the robot. A significant kind of improvement over our old OT2 robot. You can be selecting particular protocols that you want to be run. There's a guided deck setup, so that just guides the end customer with that screen on how to set up the robot. And you can be easily adjusting at time of run common kind of variables that you might want to be changing in your protocol between each run. Simple things like number of samples, elution volume, and many other highly customizable sets of parameters. So you can really kind of tune your NGS protocol for the specifics of that individual run. And that can all be done seamlessly from that on-device display. And so we'll talk a bit more on OpenTron's approach towards automating NGS library prep. And so before we kind of dive into some of the, the more kind of technical matters, we'll pass to Kinnary, I'll just give kind of just the background context of kind of how we see OpenTron's, contributing towards NGS library prep automation. And so even earlier this year, Illumina was kind of talking about their customer base and talking about particularly the needs of small and medium-sized labs, where as sequencing costs have gone down, in their view, at least with their respect to their customers, it's the first year that the sample prep costs, the labor costs associated with, and time associated with that sample prep exceeds the actual end sequencing cost. Which means that to enable small and medium-sized labs to be more kind of efficient in their sequencing activities, adoption of automation is going to be really critical to that. To one, bring down the fundamental costs associated with this activity, and second, ultimately enable greater kind of scalability of sequencing, because there hopefully through that cost savings will enable with that same budget to do so much more. And because OpenTrons is, has the most affordable NGS library prep solution on the market, it's uniquely positioned not only to, of course, provide significant benefits over manual execution, is uniquely positioned to provide significant cost savings and efficiency gains relative to other automation solutions on the market, which are oftentimes far more expensive. And these graphics above kind of give you kind of a sense of when you kind of bake in the cost of the system, the cost of the support contract, kind of the the labor savings, all the all-in costs associated with using kind of OpenTrons, it's a very, very substantial cost reduction relative to even other automation solutions for low and medium throughput labs. So with that, I'll want to jump to the next slide. I'll pass the baton to Kinnary to talk a little bit more about our the OpenTrons platform and start digging into NGS a bit more.
Yeah, thanks, John. I'll do a quick plug here for OpenTrons AI. and I'll give you some more information about our external beta at the end of this talk. But we're really trying to innovate the ways in which people write protocols and the accessibility of being able to build these applications. So stay tuned for that in a bit. So overall, taking a look at the NGS workflow, there are some really common pain points throughout the workflow. Upstream with respect to the nucleic acid extraction, the actual library prep, and the complexity and nuance there, in addition to the general length of the protocol. And then, of course, the tedium associated with quantification and dilution and normalization of those samples and pooling. So really tedious, also sometimes very arduous steps that really taken together have a huge impact on the sequencing quality of your samples ultimately. And so the OpenTrons Flex is a great solution for so many of these NGS bottlenecks, and I'll discuss a little bit about that on these next slides. So thinking about the challenges themselves, configuring and deciding on the components that are going to enable your application, getting things adapted from a manual user guide or protocol, and then optimizing that protocol so that it runs consistently and with enough flexibility to meet your needs in the lab. Those are all challenges that we've heard from customers and that we've addressed with the OpenTrons Flex. So first, the actual workstation. So we've done the work to configure a solution that is going to meet the needs of your NGS workflow. And so I'll talk a bit about the specific components, but we have recommendations that are going to enable the automation of your library prep. And then for the actual adaptation and optimization, we have OpenTrons verified protocols publicly available on our protocol library that we've recently revamped, as well as custom scripting included with all workstations. And finally, the actual usability of it, which we'll talk about the features that enable that as well, but an automated calibration system and really easy robot setup and running. Those are all components that are going to make it a lot easier for you to automate your application with really reducing the time to actually get to the science. So the components are also pretty important. So the NGS workstation on the OpenTrons Flex has either 2 8-channel pipettes or a 96-channel pipette if you're really trying to optimize for efficiency and time savings there. We have that option for you as well. And then we have the modules that you'll need to really stitch together a fully automated solution for NGS library prep. And that would include the on-deck thermocycler, the magnetic block, the temperature module, the heater shaker, and on the Flex, the gripper to really stitch together those workflows for optimal walk-away time. We also have some extension slots. So if you're optimizing towards a higher throughput or conservative use of tips, you can really expand your application. So it's a platform that can grow to meet your needs really easily. So taking a snapshot of that protocol library I mentioned, here's what it looks like. So you have several filters on the left panel here for applications. You can search, of course, and filter for NGS applications, which may be particularly relevant, Flex applications. And you'll see the protocols here listed on the right, a couple of the NGS workflows that we verified internally. And then you'll see that badge on the right for OpenTrons verified signage. And this has been a pretty popular way of identifying particular kits that have already been vetted on our platform, and pretty popular as well. We've had over 13,000 protocol downloads since the revamping of this library earlier this year. So you can download these protocols, you can configure them to meet the needs of your particular application in your lab. The other thing that's really exciting is kind of how much effort goes into making these things really robust. So just to take you inside to the team, a lot of important people standing behind me here in this picture on the right. And we have subject matter expertise internally from molecular technologies with folks that have experience setting up those secondary bioinformatics so we can get those pipelines robustly, getting the sequencing specifications that are going to be relevant to the needs of your lab for QC and for your tertiary analysis potentially as well. We also have a lot of proteomics and cellular scientists as well. And being scientists, it's really a lot easier for us to understand the needs of your applications because we have those expectations as well of how these assays need to be performing manually. And we're working throughout that application verification process to meet those specifications. So when you are able to download a protocol that's been verified, for example, here let's focus on this QIAGEN QIAseq FX kit that we've partnered with them to develop. You'll see here, which is just zooming in on the actual touchscreen or display that you'll see on the Flex. And if you click on the protocol, and you'll see that there are several, so you can have several protocols on the robot that you can use at different times. The actual QIAseq FX protocol, you'll notice a set of parameters. So if you'll see this purple button, you'll touch it and you'll see the parameters that are going to be important for you to have flexibility with for this particular protocol. And you'll see a couple of features, elution volume, wash volume, things that may or may not vary in your particular NGS protocol, but number of samples being probably a really common variable that you'll see on almost every protocol because it's often that you're running with different numbers of samples from run to run. So after clicking on any of these parameters that you would want to set in a run, you can just change the value to meet the need for that particular day. Here, if we put in 24 samples, this is a protocol format with an 8-channel pipette or 2 8-channel pipettes actually. And after that selection, you can kind of confirm the values and move forward to monitoring the run after your module, labware setup, and sample and liquid setup. So after that, you're ready to run and you'll be able to see the particular step that you're running on the robot at a given time so you can keep track and then come back to a completed run with fully constructed libraries. So that's the setup and configuration. We have really wanted to deliver on the flexibility of this platform, and that's involved a lot of verification of particular sequencing technologies. So just to mention a bit, we have done quite a bit of work with various different reagent kits here, either internally or with customers in the field to get applications running, and nearly completely agnostic of the input sample type. So we're working with DNA, RNA, panels, multiplexed amplicons. We're really trying to meet the diversity of sequence applications that can be found with scientists. We're also upstream of that. So the extraction is also important in terms of quality and compatibility with various sample types. And we've tested several internally. We will be testing so much more in the future, and I'll give a heads up about some upcoming projects. But really, we're hoping to cover every aspect of the NGS library prep workflow so that you can have ideally walkaway time at each component of the workflow. So here's what the deck looks like. So here's a snapshot of what you would see in our app if you were setting up this protocol or on the touchscreen, because we endeavor to have full walkaway time. So maybe you're just setting this up at your actual robot. And you'll see the components that we've mentioned in the actual workstation. So the on-deck thermocycler, the temperature module down in D3, which is actively cooling, master mixes and primers, heater shaker for resuspending samples, and many other incubations as well. And then our magnetic block, which is really fixed. And so the gripper is able to help us move through these applications and really deliver on that walk-away time for this particularly complex and fully configured workstation. So there's a lot here that we've done, and I'll try to show snippets of each of these aspects of the workflow, from components like cleanup, but also across the applications themselves, whole genome sequencing, targeted, PCR-free, to illustrate the flexibility of this particular configuration. So the deck setup that I'm showing on the slide previously is used consistently to perform almost all of these applications. So we're able to get a lot out of our NGS workstation, all the way up until long read sequencing. And I'll talk a bit about how we're able to support some chemistries from Oxford Nanopore, and a bit more in terms of what we have planned for additional verified protocols. So cleanup is a pretty well-known pain point for manual library prep, with resuspending beads and preventing over drying, and also avoiding ethanol contamination and so many other things. For us, on the Flex, we wanted to make sure that you can consistently get the target range of fragments that you are specifying with your left, right, what have you, in terms of size selection. And so we've done so many experiments to vary the target range, the proportion of beads, and we've been able to see really consistently accurate peak fragment sizes and just range or distribution of fragments as we get through these protocols. And we've done this in the canonical way with ladder and so many other sample types. And then, of course, with genomic DNA as well, and various libraries. So we're able to meet that need. It's something that is a bit underserved, but needs to perform consistently for you to be able to get really well constructed and accurate libraries out of the workflow. So looking again at this distribution, so we're a fully equipped lab in terms of the sequencing capacity at our HQ at Opentrons. And here, showing some genomic DNA from E. coli, we were really interested in making sure that we could maintain a level of uniformity across replicates and still have really competitive and compelling sequencing specifications. So high mapability, but low error rate and duplicate percent. So we're still able to maintain that. You're seeing the distribution of the constructed libraries with our tape station gel up here. And you're seeing a lot of the uniformity that we really work to maintain within an automated workflow. So that's E. coli. Can we consistently have that performance with something of a more varying genomic complexity? And here we had a really low input of community standards, microbial standards. So species of varying GC content in one sample. And so these were pretty error prone in terms of analysis, and yet we're able to maintain and recapitulate the expected abundance of these species while maintaining high quality sequencing specifications as well. Normalization is also, as I mentioned, can be pipetting heavy in terms of the quantification steps, the normalization of the appropriate molarity. And we've done quite a bit of this internally because of this pain point. And we've done it various ways. So qPCR, qubit, and enzymatic normalization as well, which is a really automation-friendly approach to implementing this in your NGS workflow. And so when we compare against these methods on our platform, we're able to do many different normalization methods really consistently well. So you're able to get that proportion and maintain the balance of your samples, whichever method you choose on the OpenTrons Flex. We will advocate or support a lot of the enzymatic normalization that we've been seeing. Here is a bit more from our partnership with QIAGEN, and you'll see it's pretty clear when you're starting with variable inputs, as you might be for various reasons, you're able to maintain or improve drastically the uniformity of your samples going into your pooling and sequencing ultimately. RNA as well. So we've done historically a lot more DNA, but more recently we are meeting the need of the RNA sequencing requests and inquiries we've gotten from customers. Here we've done some work with Illumina chemistries. Below you'll see some total RNA prep with RiboZero Plus using our universal human reference RNA standards. And these are really, can be really sensitive in terms of getting the incubation steps to be consistent and accurate. And we're able to do that. We have this protocol available on our library as well, if you'd like to see the steps or conditions. But again, with respect to our RNA, with respect to the percent aligned, other markers of a really high quality RNA sequencing experiment, we're able to illustrate that here. And I'll talk in a few about an upcoming product that we employed in this workflow and that we're really excited to present to you as well. With respect to flexibility, so I mentioned we really are meeting the needs of almost every sequencing approach here. And so for PCR versus PCR free, here looking at the Illumina DNA prep chemistry with and without amplification, we took a look at some of these high GC content regions to see if we're able to improve the quality with the removal of those amplification steps. And you'll see a really consistent base quality over those GC rich regions. And that's exciting. That's what we've seen manually, and we're able to maintain that observation as we automate these workflows as well. And then just to cover some very complex workflows. So there's some targeted sequencing that we've done here, showing a lung panel that we worked on. The complexity of these workflows is really notable here because they are long and we need to run them consistently, and they're with very precious samples, as they all are, but really particular sample type as well. So getting through these workflows, the specifications that we're most interested in initially were really around accuracy. Are we actually able to identify targets that are of interest in these panels? And we're able to do that, as well as reducing the hands-on time, and in some instances, increasing the throughput that you would be able to run as well. And then with respect to long read sequencing, which is a really exciting endeavor for us at OpenTrons, we have a MinION, and we've been really excited about these platforms. But the specifications we're really thinking about here are getting high yield, so getting well-constructed libraries as we prep on our platform, and then the usability of that. So are we getting a poor occupancy that would enable us to have really high quality data? And here we've done that manually. You'll see a couple of manual standards that are kind of hard to see here because they are meeting or performed better by our actual automated runs here. So we're really excited about this, and we're definitely excited about the performance of the library construction of this Oxford Nanopore genomic ligation kit. So please take a look. There's I mentioned a lot of these chemistries, but we really want to encourage you to take a look at the protocol library. You'll find protocols for all of the chemistries that I mentioned here on the left, and a few verified by members of our community. I'll also add that there are many, many protocols coming soon. So there's quite a bit of NGS application development going on now, and there are some really exciting applications that will be coming out later this year. Again, I will say that you can include it in our NGS workstation is custom protocol development. So if there's a kit that you don't see here, it's something that could very likely be developed by our field applications team as well. If you are interested in joining our external beta for OpenTrons AI, please take a snapshot of this QR code. We're really pushing the protocol development and accessibility of our platform. And in order to do that, we believe strongly that you should be able to write protocols really easily. So we're really excited about this particular functionality with OpenAI. So please take a snapshot and join us. And then lastly, I'll mention this product that is available now. So let us know if you're interested in this as well. The HEPA UV, which is really exciting, something the internal teams have been testing for some time. And we're really excited to be able to have this functionality on the Flex so that we're maintaining HEPA filtered environments for our really precious samples and libraries, of course, if you're doing DNA or RNA library prep. So with that, I will thank you all for your time and toss it to Fran for questions.
Thank you very much, John and Kinnary. Just before we jump into questions, just a quick reminder, if you want to ask a question, just go to the bottom of your screen and you'll see a little Q&A box. Type your question in there and we'll put it to John and Kinnary, and we should have plenty of time to get to your questions. So please don't be afraid. Ask your question and we'll get around to it. So, but we have a couple of questions, so I'm going to jump in. First question, the Flex system has performed even with spheroid and organoid single cells. So it's a question like, have we done work with spheroid and organoids single cells?
Yes, and if you're interested in spheroid or other cellular applications, we've published quite a bit on that. We had a poster earlier this year at SLAS on spheroid treatment as well. So we can get you that content for sure for your review. And then for single cell, yes, we're really excited. We are developing single cell sequencing applications in particular on the Flex right now.
Okay, perfect. And just as a note to Ricardo, who asked that question, I'm making a note and we'll follow up with the spheroid content after the call. So next question is about, so you mentioned obviously the enzymatic normalization. Could you just outline the process from the library prep to the normalization? Is it a separate protocol? Do you have to change the deck layout? What does that end-to-end process look like?
Yeah, that's a great question. It depends, which is always an interesting way to answer the question, but it does depend on the enzymatic approach. So Kaya, the QIAseq normalizer product actually has two components. It has one where you can just directly implement it in your workflow. So it's really just a few additions at the end of your protocol of master mixes to your samples. They also have a version of that chemistry that you can just, you would have a separate protocol. So it wouldn't be just implemented within the workflow itself. So there's a lot of flexibility there. And generally what that includes though is just an additional enzymatic step that can just be added on to the end of your NGS library prep workflow. I hope that answers your question. There also are a few protocols or workflows that you can review for both of those options, as well as so many other reagent providers.
Yeah, and we also have an application note on that as well, like whole genome sequencing paired with enzymatic normalization with the QIAGEN kits. So we can send that out as well.
Right.
Okay, plenty of time for questions, so do type them into the Q&A box. One more question is around throughput. So for I mean, I know that this is going to be another question where you answer it varies again. But Kinry, what's the throughput like for NGS workflows on the Flex?
Yeah, so for most of the standard workflows that I presented here that would generally be compatible for whole genome sequencing and similar amplicon sequencing, it's around 48. So for most of those, we're able to reach 48 samples in a fully automated configuration. That said, it really does depend, right? So if you're working on something like a targeted, maybe that's a lower throughput in general in terms of your needs, and we can meet that. But yeah, for something like PCR free or some of the multiplexed amplicon sequencing, we can get 384 samples and beyond potentially. So it depends, but for a standard workload, it's generally around 48 for full walk-away time. There are some really exciting new products that are going to enable a lot more.
Yeah, and I would also just call out that whoever, whatever your protocol is, we do have a field application science team that will provide, consultation and custom solutions depending on what your needs are. So, a lot of people like to split pre and post workflows, for example, so that will look different and potentially be higher throughput as well. So there are obviously, there is probably like not a one size fits all, but we do have a large FAS team who will provide consultation on your specific workflow. And as Kinry mentioned, we have done a lot of development of these protocols in-house as well. So we usually have some experience with whatever your chemistry is, so we can provide really good advice on how to set it up to your needs.
Right.
Okay, next question. You talked briefly about selecting parameters on the touch screen. Are these parameters that the customer can determine themselves? Are they determined by Opentrons? How do those parameters, and what are some example parameters that you can select for your protocol on the touch screen?
Right. So for an Opentrons verified protocol, these are parameters that we have specified, and normally it's pretty close to what would vary in the reagent kit itself. So we want to do justice to the chemistry. And so for Opentrons verified protocols, what you would see when you download and then upload that protocol onto your robot would be a protocol that we have specified should definitely be something that is flexible to run. If it is a protocol that you have requested, so a custom protocol, it is something that you can specify with your FAS. So you can request that, and you ask for examples, PCR cycles for NGS, number of samples, elution volumes, occasionally. Those are some really common ones. Sometimes it's fragmentation time or tagmentation time. Those are really common variables that you might want to change depending on your input quantity. And for that, you could specify that explicitly with your FAS. I'd like to be able to have the flexibility to change whatever set of variables, and they can implement that into your protocol, and you'll just see that on your touchscreen for use. In terms of doing it yourself, we do have open documentation should you be so inclined if you want to write protocols yourself. We have a really well documented API if that's something that you do want to take on. But again, you can do that via the library or an FAS.
Great. So we have just one more question in the Q&A right now, but if anyone wants to add in a question, do feel free. We still have a little bit more time, so if you have something and you wanted to ask it, please put it in the Q&A box. Otherwise, we'll just ask this final question. So the question is, if we don't see the protocol that we want on the list that you just shared, Kinnari, or in the protocol library, how can we request protocols from OpenTrons?
Yes. So I think the easiest option is really requesting an FAS consultation. So you can reach out to us. We're really, opentrons.com. And you can take a look at the components and get familiar, and then request via our contact there. For us, we're really aware of customer requests. So if there's a protocol that comes in several times in terms of requests, we're really inclined to have something really standardized for more customers to have visibility of on our protocol library. But yeah, I think the easiest route would be to contact our support and FAS team so that they can guide you through the process of getting a custom protocol.
Excellent. Thank you very much, Kinnari and John. No more questions came into the Q&A box, so I think everyone has had all their questions answered. So John, Kinnari, if there's anything you'd like to say before we sign off.
John?
Yeah, no, I mean, so thank you everyone for coming. I think we're really excited about the Flex robot in general, but in particular, its opportunity to be a really high quality solution to be automating your NGS workflows. So once again, thank you.
Oh, sorry, we have one more question. Sorry, sorry, if we can just pause to just ask this final question. If I want to just modify an existing OpenTrons protocol, is that cheaper than requesting a new one?
Modifying an existing, and I'll toss it to Fran for pricing discussion, but I do think feel free to reach out to us. We're really accessible, and I'm just my first name, Kinnari, at opentrons.com. We do want to have revisions on our existing protocol, so if you feel like there's a variable or parameter that you really want to see specified, we're really amenable to that. But I'm sure there's protocol amendments if it's something that you're using. If it's one of ours, please do let us know.
Yes, there is. Yeah, if it's really a protocol amendment, we do offer that as a paid service. And it's at a different price. It's a much cheaper price than a brand new protocol if it's being developed by our FAS team. So yeah, reach out if you have a need to revise a protocol. We can definitely do that. Oh, hang on. I'm sorry. We've got another question. Okay, one more. We've still got loads of time though. So
Sure.
what's the throughput difference between the OT2 and the Flex? These are great questions, guys. So keep them coming. We don't have anywhere else to be. We've got another 15 minutes, but we'd love to answer these questions.
Yeah, absolutely. And it's a great question, right? Yeah. So for the library prep on the OT2, our average throughput was closer to 24 samples. That was kind of our sweet spot, again, benchmarking that against standard NGS. And the thing that's really notable about the OT2 is there was no gripper functionality. So we were able to do that with either pipetting the samples from module to module from our magnetic module, or implementing a pause step to actually move the plate from the thermocycler to the other modules. And on the Flex, again, you're really doubling your capacity here. So you're closer to 48 for many of these protocols. So there is a big jump, and in terms of the robust feature set that you get with the Flex, because you have a gripper, because you have this touch screen for just changing a few parameters on your touch screen. I think you're really moving towards a full walkaway solution from the OT2 to the Flex with that increased capacity.
Yeah, excellent. Thank you so much, Kinnari. Here we go. We're still going. This is great. This is great. Keep them coming. The OT2 is more adaptable to hybridization, right? So I guess this is hype capture.
Right.
It's saying, I think the question is the Flex or the OT2 better for hybridization? I think that's the question. Ricardo, if you have any sort of clarifying comments, please type them in. But
sure.
So that's the question.
Yeah, it's an interesting question. I think for hybridization, the Flex is better equipped in general because of the capacity. You need more tips, there's more wash steps, there's more enrichment steps, more enzymatic steps on the end of the protocol. So just from a capacity perspective, you're going to have more on the Flex. The thing about the capture is that there are some chemistries that have a heated wash or a couple of those kinds of nuanced steps in that process. And for those, I think just generally the capacities are really helpful to have to fully automate those with a gripper, and moving a plate after those steps and just more deck capacity to get to a fully automated solution. So I would say that the Flex is better equipped to handle that.
Excellent. Okay, perfect. Yeah, we answered that question. So thanks, Ricardo, for the clarifying comments there. So I think, I know I've said this before, but I'll say it again. I think we're finished with the questions. So thank you so much for these questions, guys. They were it was great, and I love that we managed to answer them all. And so very much appreciate everyone's attendance, and I'll leave John and Kinnari to say goodbye as well.
Yeah, I thank you so much for attending. Again, we're really excited about these upcoming NGS applications and just the solution that we're offering here for NGS. We're really delivering on both the flexibility and increased capacity. So please feel free to reach out if you didn't get your question in. We're really excited about these applications.
Okay, thanks, everybody.